This is Lab 6, Part E. Movement of solutes across a selectively permeable membrane by Darius DeWitt and Madeleine Castagnaro. So simple diffusion occurs across a concentration gradient going from high to low, and it's separated by a semi-permeable membrane. And so an example of this would be a sodium ion moving from a high concentration to a low concentration in order to reach equilibrium on both sides of the membrane. Diffusion like this is spontaneous, which basically means that it doesn't need ATP, and it's a form of passive transport. The reason it doesn't need the energy from ATP is because there's more free energy, because the molecules are very ordered in the highly concentrated area, and it wants to be highly disordered. So that want to be disordered gives it that energy to move forward. Along with it, so basically the second law of thermodynamics states that the transfer of energy, or the transformation of energy from one form to another, increases entropy. So basically what this means is that when energy is changed, it becomes more disorganized. And equilibrium is the most disorganized state possible, which means that equilibrium has a high entropy. Chemical equilibrium, along with that fact, it is the equality of products and reactants being produced. So basically the rate of both forward and backward reactions become equal and are reversible. So once a forward reaction happens, it can still go backward and go forward even again. So the goal of this experiment is to determine the rate of movement based on size and concentration, and the concentration gradient of molecules. And so to show this, we used a dialysis tube as a membrane to establish basically a synthetic cell and create the cytoplasm and extracellular fluid. Our hypothesis is that only the smallest molecules will be able to pass through the membrane, because permeability depends on pretty much on size. So large macromolecules like starch and proteins shouldn't be able to cross the membrane through diffusion, at least, because they just wouldn't be able to. They just wouldn't be able to squeeze through such. They just wouldn't be able to squeeze through, basically. And small molecules like sulfate and chloride, which we'll be working with, are small enough so that they can just go right through the membrane. So for the procedure of part 1, the first thing that we did was locate the 25 centimeter length dialysis tubing We then folded one end and closed it off with the dialysis clip. Then as a second step, we put the bag over the stem of the clean funnel, and we filled it with 25 milliliters of the starch and the Na2SO4 solution, and that acts as the cytoplasm of the cell pretty much. And once we got that in, we squeezed all the air out, and we folded the other side and closed that with the second dialysis clip. We then rinsed it off, and we patted it down so it was dry. And by the end of this, we submerged the dialysis bag into the beaker of our solution, which acted as the extracellular fluid, and we let that sit in there for about 60 minutes. So part 2 of this procedure, we labeled test tubes 1 through 8, and we poured 20 milliliters from the beaker into a 25 milliliter graduated cylinder, and we divvied up 5 milliliters of the fluid into each of the test tubes labeled 5 through 8. We then cleaned and dried the graduated cylinder, and we removed the dialysis bag from the beaker. We rinsed it, cut it open, and poured another 20 milliliters into the graduated cylinder, and again divvied up 5 milliliters of the solution into test tubes 1 through 4. Basically, what this will do in the long run will show us it'll show us the difference between the inside of the cell and the outside of the cell. So what we then would do, we tested for starch in test tubes 1 through 5. To do that, we had to first add several drops of Lugol solution, and if starch was present, then the test tube would turn a dark blue or black color, and that meant that it was positive. However, if it only changed into a brown color, then it would be recorded as a negative result. And after that, we decided to perform the sulfate ion test on test tubes 2 and 6. So we added several drops of 2% BaCl2 solution, and that showed us, well, if the sulfate ions were present, then it would turn a white color, and that would be a positive test result. However, if it didn't turn white, then it would be a negative test result. And so the next 2 experiments, we tested for both chloride and protein, basically doing the same thing as the other 2 tests before them. However, for chloride, we used silver nitrate, which again turned a white color. And for protein test, we had to use the Biuret reagent, which had a bright blue color. So if it only turned a bright blue color, then it would still be a negative test. So on the picture to the right, you can see test tube 1 is over here, which was positive, and test tube 5 was negative. And you can tell this because test tube 1 turned the dark blue color that we talked about before, and test tube 5 did not. So basically what we concluded is that starch was too large to cross the membrane, so it was only found where it started out in, pretty much. When we tested for sulfate, we found both test tubes 2 and 6 were positive. As you can see, they both have that white color to them. And so that proves that sulfate did cross the membrane and therefore is small and permeable. We then checked for chloride, and again both are positive and a white color, which leads us to believe that it is again a small molecule that is permeable and it can cross the membrane. The last test that we did was on we're to find proteins. As you can see, test tube 4 was negative because it only turned that bright blue color, while test tube 8 was positive because it had a purple color. And so what this proves is that proteins are very, very large because they could not cross the membrane like our hypothesis stated. We knew that both proteins and starch were larger molecules, were large macromolecules actually, and would most likely not be able to at least diffuse through a membrane. Unlike, however, chloride and the sulfate ions. So that concludes the experiment, and Maddie will have the second part to that.